Hi everyone, I'm Josemary on behalf of GenScript, and welcome to our expert interview series. Today we are interviewing Dr. Kristian Hargadon, and he will be telling us more about gene analysis for cancer. So, Dr. Hargadon, can you please tell us more about yourself and your research interests?
Yes, so I'm actually a professor at Hampden-Sydney College in Virginia, interestingly enough, where I did my undergraduate work. And one of the things that I so appreciated kind of coming from a small school was the opportunity to get involved in research as an undergraduate student, where I began my interest in cancer biology and tumor immunology. And so from Hampden-Sydney, I went to the University of Virginia to study in a couple of labs, a tumor immunology lab run by Victor Engelhard and a viral immunology lab run by Thomas Bracciali, in both settings where I really received excellent broad training as an immunologist. And really, when I came back to Hampden-Sydney in 2009, brought a lot of the experience I gained there to develop my own research program, focusing primarily on melanoma research, but looking at other cancers as well. And in addition to wanting to understand more about tumor immune suppression and cancer immunotherapy, also really looking at things from a genetic perspective and genetic drivers of cancer progression and genetic influences on immune suppression, tumor escape, things of that nature.
That sounds very interesting, and I'm sure you've had a lot of applications for the research with genomic analysis on highly tumorogenic cancers, and have had a lot of important findings for the medical community. Can you tell us more about your research with gene editing specifically and how you use it to target highly aggressive cancers such as melanoma?
Sure, yes. And you mentioned this idea of genomics. It's really just become such a driver of what we do with, data emerging like the Cancer Genome Atlas and these other large collections of patient data where we're learning so much more at a genetic level about cancer. For me, that's been really useful. Again, at a small undergraduate institution, I can now look at that data and take what we're seeing, what is being seen in clinical settings, and say, okay, how can we study this better in a mouse model, which is really what I work in. And so we're seeing, certain genetic factors that are either mutated or genes that are abnormally expressed in patient tumor samples, correlating that with patient outcome and saying, okay, why is gene A associated with aggressive disease? And so one of the genes that we've become particularly interested in is a gene that encodes a FOXC2 transcription factor, sort of a genetic regulator in and of itself. We've seen that can be overexpressed or abnormally localized in patient samples. And so now we use a melanoma mouse model. We've got tumor cell lines that grow with, as you said, different tumorogenicities and seeing upregulation of FOXC2. So the question for us became, well, can we use genome editing to disrupt this gene, knock out its function, and then see how that affects cancer growth, response to therapy? These are some of the things that we're going to be looking at going forward. But this was really the work that was being highlighted by Molecular Cloud and GenScript in the recent distinguished genome editing competition, where we were disrupting a particular location within the DNA binding domain of that transcription factor and seeing how it impacts tumor growth.
And what were some of your findings with the FOXC gene knockouts?
Yeah, so it was really interesting. Just in and of itself, the tumor certainly grew with slower kinetics when we implanted that into animal models. But what was also really interesting was when we took a genomics approach to study the knockout versus the wild type melanoma, we saw major differences in expression of some key genes that are involved in, say, interferon responsiveness, which we know is important for immunotherapy. We saw an upregulation of genes in the knockout or in the wild type tumor with regard to the oxidative stress response, which we know is important in resistance to chemotherapies and some targeted therapies. And so now we're really interested to explore going forward what role, if any, is FOXC2 playing maybe in resistance to some of the cutting edge therapies for cancer, immune therapy, targeted therapy, things of that nature.
So in your genome editing design, what kind of gRNA tools helped to facilitate this genome editing?
Yes, we use the GenScript, the publicly available guide RNA design tools. So it was very easy to use, basically taking the gene sequence, the known sequence for FOXC2, and trying to identify within that sequence particular regions that we wanted to target, as I mentioned, in that DNA binding domain of that gene is really where we were interested in targeting. And so in doing that and finding an appropriate guide RNA that was likely to have minimal off-target effects, these were all considerations that we had, and that tool allowed us to very readily design a robust guide RNA to target that gene.
And so in a future where genome editing becomes something that can be used to treat cancer in a streamlined fashion, or something you can walk into a doctor's office and sort of have that individualized gene therapy, what would that sort of treatment process look like?
Yeah, it's interesting. It depends on the actual application, I think. I mean, I think in and of itself, just genome editing, I think the reason that Jennifer Doudna and Emmanuelle Charpentier received that Nobel Prize, certainly for therapeutically what it might mean, but also just what it means for the scientific community in general, whether it's disease-related research or not, the ability to manipulate genes, knock genes out, alter, coding sequences or single bases with this technology. We're going to learn so much biology that I don't think we can even begin to fathom, where that field will take us. Now, in terms of cancer therapy, certainly, there are the ideas of, well, here's a mutated gene, let's edit that so that we could restore normal function. I think some of these are goals to shoot for. I think, in the more immediate future, there are challenges in delivery. How do we deliver, a therapeutic to all cancer cells at the time someone's diagnosed? Though I'm seeing some really interesting work in animal models, preclinical studies, targeting these and showing some effect. So certainly that's something I think is delivery of these agents becomes something we understand better, and how to minimize off-target effects. That may one day indeed be a great reality. I think in the more immediate future, there's some really exciting work out of Carl June's laboratory, University of Pennsylvania, where they're engineering T cells for cell-based therapeutics. So now you're getting away from having to worry so much about, just delivering something to a patient, a genome editing drug or delivery system to a patient at large, where you're trying to restrict it to the tumor. When you're using cell-based therapeutics, on the other hand, you can manipulate those outside of the patient. And so they've done some really exciting work with T cells, redirecting them to tumor antigens while also editing out, for instance, the PD-1 inhibitory receptor that's such a hot target for immune therapy now. And so they've seen that's in phase 1 trials. It's shown to be safe and they're getting durable expression of those transgenes in the transferred T cells. So I think that's a really exciting study that's come out, and I think that's going to open up a lot of possibilities in that field going forward. Chimeric antigen receptor therapy, all of those are really exciting.
Well, Dr. Hargadon, thank you so much for meeting with us today and sharing more on your research with genome editing, highly aggressive cancers such as melanoma, and where the future of genome editing is heading. And congratulations again to your team for winning the 2021 Distinguished Team Awards from Molecular Cloud, and stay tuned for the next expert interview.